There's a lot. We'll look at examples later on where it's like measurements in food. In those cases, it's usually on like a per gram basis or something. So we want to make sure to have those units in there just so that we're aware of what it is. There you go. The second issue, and I noted this on those that had it, was the misinterpretation of what the question was asking. So it wasn't that what is the one that grows at pH 4, 7, and 10. Those were all the same bacteria. Based on the growth, what was it? So just take a look at that and look through it afterwards, because that's one microbe that's been grown in those 3 different environments. What we're looking for is where does it grow best, okay? So those were the only 2 notes I wanted to make there. The other thing I wanted to mention, so here are the objectives for today. So we're going to continue to focus on microbial growth in oxygen requirements just for a little bit. I want to add one more layer of depth beyond what we've talked about so far. I want to briefly cover spores. We're not going to spend a lot of time with them, but they are not only medically important, but also important in cases of food safety. We'll talk about 2 of the methods for measuring growth, and then we'll spend a lot of time working on calculations. And I do want to take some time to cover the pluses and minuses of a direct method of measuring growth versus an indirect method, because there are some benefits and drawbacks of both of them. So here's where I want to start. With a problem. All right. You can do these. This is said to be done in groups of 4, but you can do it in groups of 2 if you want to. I at least want you to talk with somebody else on it. So it says, obligate aerobes grow only in the presence of oxygen. Can Mycobacterium tuberculosis, an obligate aerobe, perform anaerobic respiration? Justify your answer. This in some ways is review. But I also want you to start to think about, where does it grow in a thioglycolate to? And then, after you've done that, I want you to tell me what you think would be the results of the 3 other tests up there. Would it be catalase positive or negative? Would it be positive or negative for phenol red glucose? And would it be positive or negative for fermentation of glucose, sucrose, or lactose in a triple sugar iron auger? And would it produce hydrogen sulfide? Okay. So this is going back to metabolism, but there is an importance here to doing this. So I'll give you about 3 minutes. Go. All right. So there's a thioglycolate tube. Again, we'll work with these tomorrow in lab. But let's just start with obligate aerobe. What does that mean about the organism's oxygen requirement? Brendan? It requires oxygen. Therefore, what type of metabolism is it going to perform? Just aerobic, nothing else. Where is it going to grow in a thioglycolate tube? Yeah, right at the top. So Mycobacterium would grow right here. Positive or negative for phenol red? Glucose phenol red. Negative. Triple sugar iron? Negative, negative. Catalase? Positive. What is catalase? Brendan? Yeah, so it breaks down hydrogen peroxide. It's the fastest known enzyme, or the enzyme that works the fastest that we know of. And that's where I want to start this conversation, is talking a little bit about this extra level of protection. So for example, what differentiates an aerotolerant anaerobe from an obligate anaerobe? We said that both of them can perform fermentation. They both can perform anaerobic respiration. But there's a difference there, in that obligate anaerobes never grow in the presence of oxygen. Aerotolerant anaerobes do. So why is that? And it really comes down to the presence of some different enzymes. and I know that this was in the lecture, but I just kind of want to review this real quick. So in metabolism, particularly aerobic respiration, there is the chance to produce what we call free radicals, or radical oxygen species, or reactive oxygen species, ROS. And this is what I want you. I don't want you to know necessarily how these get created. Just know that these are a natural product of oxidative of the electron transport chain and aerobic respiration. What we find is that most organisms that can grow in the presence of oxygen have two enzymes, or at least two enzymes. One that's called superoxide dismutase, and the other that we've tested for called catalase. Superoxide dismutase takes radical oxygen species, breaks them down into hydrogen peroxide and oxygen, and then catalase takes those hydrogen peroxides and breaks them down into a couple of molecules of water and oxygen gas. Peroxidase, on the other hand, is a different one. It takes hydrogen peroxide and hydrogen ions and breaks it down into water. I'm not all that concerned with you understanding the chemistry behind this, but these are protective measures against oxygen. And so when we consider the growth of a microorganism, we also need to think about, based on its oxygen requirements or where it grows, does it or does it not have these particular enzymes? So if we take a look at some examples of thioglycolate tubes again. 1, 2. What I'm going to do real quick is I'm going to draw growth in each of these, and then as a review I'll have you identify what's its, based on that, what do we call it? So is it an obligate aerobe, obligate anaerobe, yada yada yada, and does it or does it not have catalase? Would you predict it to, or would you predict it not to? All right, there are the 5 different tubes. Go ahead, take 2 minutes, 3 minutes. All right, let's go through these. Let's at least just start with, let's assign each of these to one of the 5 tubes. So our microaerophiles, that has to be A, and we said low amounts of oxygen. They perform aerobic respiration, though. They still require some oxygen to live. So that's A. How about our obligate aerobe? D, good. Facultative? Okay, what's the giveaway here? Yeah, but usually grow better in the presence of oxygen than the absence. If you had said C, you know, that would be very close, but there is that fine distinction that we said. It can be sometimes hard to tell in person. Obligate anaerobe, and then aerotolerant would be C. Perfect. Okay, so the next question is then, do these have catalase, or do these have those types of enzymes that protect them against oxygen? We've already defined that for our obligate aerobes that yes, indeed they do, because they grow in the presence of oxygen and they grow very robustly in the presence of oxygen. What about our facultative anaerobes, E? Yes. Yeah, they do as well. What about microaerophiles? The answer would be yes, but usually this is like a yes, but. Yes, but they may not have all of the enzymes that are necessary to grow at the highest concentrations of oxygen. or maybe the expression of those enzymes is fairly low, which allows them to grow okay in low concentrations, but they can't handle very high concentrations. So I'd say a yes for this, if you were looking for just a simple yes or no answer. How about our obligate anaerobes? Nope, definitely not. Aerotolerant. Yeah, why? It doesn't matter if oxygen's there or not. Anybody watch the honey badger video over the weekend? Everybody's laughing because they didn't watch it. I'm greatly embarrassed. So aerotolerant anaerobes definitely do, and that is one key distinctive feature between the aerotolerant anaerobes and obligate anaerobes. They both can perform fermentation. They both can perform anaerobic respiration. But those aerotolerant ones have catalase. They have superoxide dismutase, which allows them to grow in the presence of oxygen. It makes them completely distinct from those obligate anaerobes. Yes, so it doesn't use the oxygen, but it's able to break down any of those reactive oxygen species that may form. Yeah. Any questions with this? So again, that thioglycolate and these distinctions based on oxygen needs and requirements, etc., tell us not only about the metabolism, but they tell us about these particular enzymes. Okay? Let's look at a slightly different example here. There are some bacteria, some obligate anaerobes, that actually will, well, I guess I gave away the answer here already. The question was going to be Clostridium sporulates in the presence of oxygen. What does this tell you about it? And it tells you that it doesn't like the oxygen. It doesn't survive that harsh environment. So really the question that I'm asking is, what are spores? What is a bacterial endospore? Ashley? Yeah, so a couple of the key features of those endospores are these really thick coats on the outside. The image on the right here is a bacillus. I'm trying to remember which exact species, but it's in the Bacillus genus. Here is that spore, and then this is still the vegetative cell. I'm not going to ask you guys to know a lot about spores. I'm not going to ask you necessarily how they form. But what I wanted to point out is they're called endospores because they form inside a mother cell. And then this mother cell in the environment can rupture, releasing that spore. Now, spores form in response to environmental stress. Not all bacteria form spores. In fact, does anybody recall what they're limited to? Well, what kind of bacteria? Sorry, gram-positive, and usually the other designation is it's bacteria with low GC content. That's not really that important. But the major ones of medical interest here are Bacillus, the genus Bacillus, and the genus Clostridium. So if you've heard of Clostridium tetani, Clostridium botulinum, Clostridium difficile, Bacillus anthracis, for example, those are 4 medically important examples of bacteria that have the ability to sporulate. Spores, because of this thick outer coat here, are protected from a number of different chemical treatments. They're resistant to bleach. They are incredibly resistant to 70% alcohol. Actually, what we do in lab to spray down the benches is not enough to kill a spore. 70% ethanol has no effect on them whatsoever. They are heat resistant. Some can remain viable or still quote unquote living at 100 degrees Celsius for 2 hours. And so when we sterilize liquid media in the lab, something that we'll begin to talk about on Wednesday, We actually have to go above that temperature point. We usually go to about 120 or so and hold that temperature for about 15-20 minutes to kill spores. Spores typically are dehydrated. The more dehydrated it is, the more resistant it is to environmental stress. And we have enzymes and proteins that help to stabilize DNA, help to stabilize protein structure, so that when these germinate, they can form fully viable, what we call vegetative cells, rapidly growing cells. I was looking for stuff on this yesterday, and I read an example where from a piece of amber, some scientists down in South America isolated a spore from the stomach of a bee, preserved in amber, 25 million years old. They were able to actually get it to germinate. So these can survive for incredibly long periods of time. They are metabolically inactive. They're dormant. It's kind of like, it's analogous to what a seed is for a plant. That once the environment gets harsh, the plant produces seeds as a way to ensure that progeny, that its life cycle or its progeny will live on afterward. Okay, that's all I wanted to cover for spores right now. We'll come back to them as we talk about some of these different physical and chemical methods for controlling growth. But that's about all I wanted to make sure to control or mention. The other term I brought up was persisters. I really don't want to talk about them, so we'll just skip them. But it's kind of like the gram-negative version of a spore. But those cells are not dormant. They are still actively growing. They just grow slower, and nobody really understands how they form. So we'll skip that for now. Where I want to move into is talking about growth. There are a number of ways to measure bacterial growth. We use primarily two different methods, one that's an indirect method and one that's a direct method. The indirect, using spectrophotometers. And this is what we will do in labs starting, we'll run an experiment Tuesday, analyze the results on Thursday, looking at the growth of your unknowns using spectrophotometers. The second method is what we call a direct count, and that's what the homework assignment was really getting at, using serial dilutions and then plating out bacteria to determine the number of colony-forming units per mil. How does a spectrophotometer work? Or if somebody was to describe how, when we measure bacteria using a spectrophotometer, what are we actually measuring? It's optical density. And the optical density is really the same thing as, it's the same units, it's measured the same way that you would measure absorbance, but bacteria don't absorb light. Light bounces off of them or around them. They block light from getting through. And so this optical density measurement, as there's a greater concentration of cells within the culture, the optical density increases. What are some potential pluses of this? Imagine this in comparison to plating and counting plates. What's a potential benefit of this? Incredibly time efficient. Actually, I don't know if anybody knows Bill Bull, but him and I were running an experiment on Friday, and we were able to measure the growth of E. coli with bacterial viruses from 6:30 in the morning to 7:30 at night every single hour. And we got a really nice growth curve out of it. Could do that in one day. Versus imagine trying to plate every single hour. Serial diluting and plating every single hour would be painful. It would take literally all day to do, and as soon as one time point is done, we would have to start again. What else is potential benefit here, or potential drawback? Let me try to draw. We'll play Pictionary for a minute, and we'll see if this helps with identifying a potential drawback. So imagine two tubes with different bacteria in them. And let's say that each of these tubes have 5 bacteria in them. Anybody see a potential issue? Absolutely. Absolutely. Size does affect the optical density here. And so what this means is that we can't compare results between different species per se. That oftentimes we have to, if we wanted to know what concentration of cells in CFU per mil correlates to an optical density, we'd have to calculate that for each different species of bacteria that we're working with. The other thing, and I think some of us have even noticed this in lab, some bacteria actually shrink as they go from log phase into stationary phase. And that change in shape can also affect optical density measurements as well. So one of the downsides here is that optical density measurements do not account for those size differences and changes that occur within cells. But really quick, really easy to do. Direct plate counts on the other, and the last thing here I wanted to mention is that these are just, we say that they're indirect measurements because we're not actually counting the concentration of the bacteria. We're just getting a rough estimate of the concentration based on that optical density. But these are bona fide measurements in any scientific paper that deals with bacteria. These are acceptable results. Now, plate counts, on the other hand, highly specific, oftentimes highly repeatable. For those folks that really like quantities, this is a great way to observe growth or death. Downside, time-consuming, incredibly time-consuming. One other thing I want to point out here, actually, that's a difference between the two of them. Spectrophotometers have what we call a limit of detection. In other words, what we mean by that is that it can't, oftentimes in E. coli world, we can start to get an optical density reading at around 3 times 10 to the 7th cells per milliliter. Anything below that would give you the exact same reading of 0. And so what that means is that you can only observe optical density measurements, or you can only see changes with those at higher concentrations of cells. Plate counting actually can be much more sensitive. So for example, from a culture of bacteria, if we plate a full milliliter of culture onto a plate and we detect one colony, we can assume that our concentration of cells is 1 bacterial cell per milliliter. It's a much more sensitive method than is measuring optical density. So sensitivity is greatly increased. I'm going to pause here real quick because I know that the calculations went well, but that doesn't necessarily mean that folks understand what the hell is going on here. So I wanted to find out what questions you guys have about serial diluting and plate count before we move ahead. So I'm gonna pause for a minute, give you guys a second or a few minutes, a minute or so to think of questions, and then we'll move on if we need to. But otherwise, I'm willing to answer anything here. So typically what we do here is the first time you ever run an experiment where you don't really know where to begin, you would do a full series like this, and usually the rule of thumb is count the plate that has between 30 and 300 colonies. So you may see these 3 plates, but don't count them all. Oftentimes people it's a waste of time to count every single plate. Just choose that concentration that gives you between 30 and 300, count that and do your calculations. If you were to do this again down the road, I would say don't plate this one, don't plate this one. There's way too many colonies. You're not going to get an appropriate count for it. What I usually do is experimentally, whatever plate I got my count off from last time, I make that the middle in a series of 3. So usually I do we'll plate 3 different concentrations, so that if it's a little bit less or a little bit more than last time, I've at least covered my bases, so that I can actually get some measurements out of it. One downside of this is that not only is it time consuming, but it takes overnight to visualize these results. The upside of the optical density measurements is it's real time. You actually never have to take the cells out of the plate or out of the culture to do those measurements. Excuse me. So it can be done in real time pretty darn quickly. Other questions on this? Okay. Let me see what I had written down next for us to do. I think I want to do some practice. I want to take a few minutes calculations, just so that we're all on the same page there. And then we'll come back on Wednesday and we'll start our discussion with the first couple of questions on here, because I think that's a really good starting point. So I got asked a question. Let's do this first one because this will actually help with a couple of things. I got asked the question, if I'm saying a dilution of 10 to the negative 1, with that, another way to write this, and sorry, I shorthand this, but it's 1 times 10 to the negative 1. In other words, this is 0.1. Okay, if you were trying to convert this to a decimal point, this is the same as saying 0.1. Now, I like working in exponential values. I am not holding you to have to do that. I would say just find what works best for you. But I'm also going to say that you should probably know the conversion with these as well, or how to at least go between exponential and the other, just in case problems pop up. 100 microliters is how many milliliters? 0.1. 0.1. So it's 1,000 microliters for every 1 mil. So this is 0.1 mils. So again, our basic equation is CFU divided by dilution times the volume. That volume is the volume you plate. Dilution obviously is dilution. So we have 267 CFU divided by 0.1 times 0.1, and that should equal out to, if I'm not mistaken, 26,700 or 2.67 times 10 to the 4th CFU per mil. Make sure to get those units on there. Okay. If you've got that, great, you're good to go. If you don't, come talk to me or continue to practice on them, and we'll go over the rest of these later.